So in this video, we're going to talk about meiosis. And meiosis, the whole point of it is to half the number of chromosomes, okay? So prior to meiosis beginning, we actually have duplicated our chromosomes. So we actually have, just like what happens at the beginning of mitosis. So we've actually done DNA replications. We have 2 sets of chromosomes, essentially, okay? And in prophase, what actually happens is kind of very similar, in prophase 1, I should say, very similar to mitosis. The chromosomes condense. And we have our little mitotic spindle forming, or meiosis spindle in this case, is starting to form, and we're going to, the other thing that happens is my homologous chromosomes pair. And so what we mean by that, or homologs pair, is that you actually have 2 chromosomes, one you got from mom and one you got from dad. And so we're going to kind of draw those like little X's, and they're going to partner up, and you can draw them wherever randomly. They're going to partner up, and they're going to pair up. And so chromosome number 2 is going to find its match on chromosome number 2. And chromosome number 4 is going to find its match with chromosome number 4, okay? So they're all going to find its match, and so I'm just kind of representing them in blue and red. And something else is going to happen. We're going to start to see, and I kind of drew it in this one here. You can kind of see how I made these 2 legs cross over, and we'll zoom in on that. But something that's called actually crossing over is going to occur. Begins between non-sister chromatid. And then in metaphase, just like before, we're going to have our chromosomes line up along the metaphase plate. And so I'm going to draw mine, and I'm actually going to do this in a little bit of a purposeful manner with my blues and reds, because it doesn't matter which side they line up. It's actually quite random which side they line up on. And you can actually you can do little red tips on some of them. I'm going to do a little red tip there. So that means that this one will have a little blue tip on it. And I'm going to do a little blue tip on this one, so that means that this one will have a little red tip on it, okay, because they crossed over. And they're actually still going to probably be attached to each other at this point. So in this one, the chromosomes line up, okay? And then in anaphase, they're going to begin to pull apart, okay? So the homologous chromosomes move apart. Okay, and so we're going to see, here's my little blue one, and here's my blue one with the red tip, and then I'm going to have my red one with, and this one's going to have a little red tip, and then I'm going to have this guy like this. Isn't this so much fun? And it's got a little red tip. Okay. So now they're starting to move apart. And then by the time they get to telophase and cytokinesis, we're going to have that cleavage furrow form, and I'm going to end up with 2 cells. And they actually are going to kind of regather again. And so we're not going to really have, I'm just going to kind of draw them like this. Just chaos, okay? Okay, that was just because my pen's crazy. But like, they're going to recondense, okay? And so cytokinesis occurs, obviously, in telophase 1, and chromosomes gather in nuclei. Now, at this point, we really have just ended up with non-homologous chromosomes, or spread apart. And so what we need to do is we need to split them again, because we want to half the number of chromosomes. So that's where we're going to do the same process essentially over again, okay? But replication has not occurred. And so in prophase 1, the chromosomes will condense. Okay, so we're going to start to see that we have these guys here. And for just the sake of video time and all that kind of stuff, I'm just going to have one of them have been crossed over, this guy here. And so then I need a little red on this guy here. Okay, a lot of them can have crossed over at this point. And And then what's going to happen is they're going to line up at the metaphase plate. Just like before. So we're going to have, here's my little red one, and my blue one, and my blue one, and we got to put a little tip on this one. And then here's my blue one. We're going to put the little tip on this one. It would be on this side. Okay, so that's where this tip is from crossing over in that happened in prophase 1. And so now in anaphase, the chromosomes will spread apart. So they will separate, okay? And actually, really, I should actually say not chromosomes, but I should really use the word chromatids, because that's really what they are at this point. And so, like, if you were to just separate this one here, I would end up with this guy, that's this side, and this guy, and then this one. And then on this side, I would have this one and this one. And then I would have this red one with the blue tip end, okay? And then if I were to do this one, it'd be kind of similar. I'd have this one separate like that, and I'd have the red one separate like that, and then I'd have the blue one separate, and then I'd have one with a red tip, okay? And so then in telophase, what I have now in cytokinesis, I will end up with 4 separate daughter cells that are haploid. They only have half the number of chromosomes, okay? So I have 4 haploid cells. And I'm going to kind of just, I'm going to zoom in just for a second on the one, this one here that's going to form from these ones, because it's going to end up with 2 red ones, but one of those red ones has a blue tip, and then this one. We call this cell a recombinant cell because it's got part of a chromosome from a mom, the red part, and then a part of a chromosome from a dad, the blue part. Okay, so that's what we call a recombinant cell, because it has that mix from crossing over. This one, it would be called a parental one because it has no crossing over chromosomes. And same with, I'll draw this one, the other one. And these guys are going to like end up decondensing these chromosomes during telophase 1, or as it moves into normal, become a gamete that way. Okay? Some other words that are kind of associated with this whole thing. So let's kind of just redefine crossing over. It's when the ends of chromosomes switch places. And some of you are like, why do they do that? It's really a lot of these things on this page is to create genetic diversity. Okay? Places. Whatever. So, okay, they're going to switch places in the spots. So the little tip ends. So here's my one homologous chromosome, and here's my other one. And usually they kind of actually lay on top of each other like that, and they actually break off, and then they end up looking like, you'll end up with one like that looks like this with a little red at the end, and then you'll end up with one that looks like this with a little bit of blue at the end. Okay. So that's the deal with that. And what they create is they create recombinant chromosomes, okay? So a recombinant chromosome is a chromosome that results from crossing over. So really the whole goal of meiosis is to create this genetic variation. So genetic variation really is just the variation of genes in a gene pool. And it's really due to a few things. One is crossing over. One is independent assortment, which I will talk about in a second. And the other is random fertilization, which I will talk about too. So those are kind of the things that create this genetic variation in a cell and really in life. So this law of independent assortment is really about this, is that chromosomes line up independently. Line up independently of each other. In metaphase 2. Okay? Or I'm sorry, metaphase 1. So let's go back to this picture. If you go back to this picture, I drew the one blue chromosome, the one that let's say this is an egg, it's going to be going to turn into egg cells. So this is a mom that I drew this one that she got from her dad on this side, but on this side is the one that she got from her dad. They lined up independently of each other. It's not like they talked and they're like, oh, all the ones from dad line up on the right, and all the ones from mom line up on the left. Or the ones with the pretty genes line up on the right, and the ones with the ugly genes line up on the left, or the good genes. It's all random, and it's very independent of each other. So what that does is it creates every single egg cell that's produced is random. There's 23 chromosomes in your body, and so how they line up, the one that you got from mom and the one that you got from dad, and how much they crossed over is all random, and so that creates more genetic variation. That's why you don't look just like your sister or your brother. You look kind of like your sister or your brother, because you might have gotten this chromosome from your grandpa and your sister got this one from your grandma instead, because that's how it just lined up and worked out that way. Okay, so it's all kind of random. We'll talk more about that as we go through this whole genetic thing. The other thing, it's random fertilization, and so which sperm bonds with which egg is random. So it's not like the sperm's like checking out the egg and it's like, oh, you have the gene for, I don't know, red eye, like brown eyes. I don't want to have brown eyes. I have the gene for blue eyes, and that's recessive, and I don't want to make a brown-eyed baby. And so, like, it's not like they have a conversation, or there's any kind of proteins, like a cell conversation would involve proteins, right? It's not like there's any kind of cell proteins that are like talking about what they have and what they don't have. It's just, it's random, and so it's random what egg ends up with what sperm. And if you think of a male human being produces 100 million sperms in one day, does meiosis 100 million times in one day. Females do it significantly less. We only do it a few hundred times in our lifetime, but still, that's a few hundred options of different babies that you can create just from her eggs, plus the 100 million sperm a day option. There's a lot of variation that's to be had in a population just from two people. And so that's why you end up with children that look very different from each other and act very different from each other, all because of these things. So that's kind of what creates genetic variation in our population. And it's really, a lot of it has to do with this meiosis process.